Hello and welcome to the August 2025 Around the World seminar. I am Terry Flott, current president of the American Society for Gene and Cell Therapy, ASGCT, and I am very pleased to co-host this session and to welcome you from all across the world who are watching this webinar. With my co-host, Dr. Jennifer Adair, we have chosen to focus on ASGCT members who are engaged around the world to globalize the spread of gene and cell therapies. In recent years, a growing number of American gene and cell therapy researchers have become deeply engaged with colleagues from various low and middle income countries and worked with them to open access to gene and cell therapies to more patients in need. Hemoglobinopathies in general and sickle cell disease in particular disproportionately affect people in areas where malaria is endemic, typically sub-Saharan Africa and South Asia. As new gene and cell therapies for sickle cell and thalassemia have been developed, researchers have been alerted to the fact that healthcare systems in the affected countries may have gaps in the resources required to deliver cell and gene therapies to the patients who need them most. We will hear today from 3 ASGCT members who are working to overcome these resource barriers. Let me now introduce the co-host for this session, Dr. Jennifer Adair. Dr. Adair is professor and vice chair of genetic and cellular medicine at UMass Chan Medical School. Dr. Adair.
Thank you so much, Terry. Just checking that you can see and hear me okay. Can I get a quick message in the chat letting me know that you can see and hear me okay? Perfect. Thank you so much. Hello, everyone, and welcome today. To ensure enough time for our speakers' presentations and Q&A, please note that the program will go over the allotted hour time frame. If you cannot stay for the entire program, we invite you to revisit today's program on demand through your registration page or look for an email with the program's recording. We have 3 presentations today, including myself. Our first speaker is Dr. Ambrose Wonkum. He is a professor of genetic medicine and the director of the McKusick-Nathans Institute and Department of Genetic Medicine at Johns Hopkins University School of Medicine in Baltimore in the United States. He also serves as the director of GeneMap, Genetic Medicine of African Populations, and is the immediate past deputy dean for research at the Faculty of Health Sciences, University of Cape Town, South Africa. Dr. Wonkum completed his MD training at the Faculty of Medicine and Biomedical Sciences at the University of Yaoundé I in Cameroon. He pursued a thesis in medical sciences and cell biology at the University of Geneva in Switzerland and earned a PhD in human genetics from the University of Cape Town, South Africa. He also received advanced training as a medical geneticist at the University of Geneva, one of the most reputable centers for genetic medicine, and has practiced genetic medicine in European, African, and American contexts.
Yeah, good morning, good evening, everyone. My name is Ambroise Wonkum. I'm a professor of genetic medicine at the Johns Hopkins University School of Medicine. I'm also the director of the Department of Genetic Medicine. It is my privilege to you today to convince you that we need to invest in African genome variation or African genomes variation to improve therapeutic opportunity for sickle cell disease. We were also very pleased on December 8 when it was first announced a FDA-approved gene editing for sickle cell disease in 2003, 2 years ago. And Victoria Gray, this mother of 4 on the slide here, was the first courageous person to have submitted herself to the editing for the cure of the condition of sickle cell that she had. This is a major achievement, but there are still a few elements or questions or observation or queries that we need to follow for the years to come as we have more and more people benefited from that therapeutic modality. The first is that the bone marrow conditioning, would that lead to any risk of chronic hematopoiesis and potential risk of cancer in those patients? Those are one question that we need to follow for the next 10 years, perhaps 20 years. The second is that the current modality of gene editing, with the goal of increase the level of fetal hemoglobin, might not necessarily achieve the goal of reducing end organ damage that we observe in sickle cell. We know, for example, that in our cohort from Africa, from Senegal, most patients have high level of fetal hemoglobin above 20%, but they still have some kidney dysfunction. And a similar observation have been found in sickle cell disease patients from the Middle East, for example, in Saudi Arabia. A third observation and queries that we still need to follow up is the potential off-target effect of the editing. We know that the current target for BCL11A that was the focus of gene editing to cure sickle cell disease, there is a very common allele in that target that is disproportionately frequent in population with African ancestry, up to 4.5% minor allele frequency, suggesting that one patient out of 20 potentially can have an off-target effect specific to patient with African ancestry, and we need to follow up on that. But today I will focus on the need for us as scientists to look for more target for therapeutic manipulation. And I argue that the African genomes are the best place to invest in new therapeutic modality, exploring new therapeutic modality genome-wide for sickle cell disease. There are three major reasons to invest in African genomic variation. The first is that we are all African. Humanity evolved in Africa about 300,000 years ago. Stay in Africa and move out of Africa about 70,000 years ago. That suggests that many variations that move out of Africa with population with European ancestry and population with Asian ancestry represent only a small fraction of genome variation of humankind, suggesting that a lot of the majority of human genome variation never actually move out of Africa. The second reason is the ecology of Africa. The north-south axis of the African continent, as we can see on the panel C, is associated with different type of environment, and these environments are associated with different type of environmental direct exposure, like the sun exposure, direct type of infectious diseases, and direct type of food. Direct environment, infectious disease, and food are mode of natural selection. I will speak to a few examples later. And because humanity stay in Africa for so long, 300,000 years, with a lot of wave of migration out of Africa, within Africa, out and back to Africa, as shown on the panel B, the African genomes is by far the most diverse of humankind. If we speak, for example, for ancestry, this is some recent data. The panel on the left, at least from on the left of my screen, show the evolutionary tree of the Y chromosome, the chromosome that is found only in male. And the roots of that tree is found in Africa. That means the oldest Y chromosome of humankind. That group is called haplogroup A00, and it's only have been described in Africa, specifically in Western Cameroon. It happened to be a country where I was born, suggesting that in my DNA I might have that A00, also suggesting that for you all listening to me today, I am your ancestor. Again, in Africa, there have been a population of humanoid that never move out of Africa, unlike the Neanderthal that move, for example, that live in Europe, or the Denisova live in Asia. On the left-hand side, there are two people here. The first one on the top is Lee Berger. He's an anthropology geneticist from Johannesburg in South Africa. The one in the bottom is Cyril Ramaphosa. He's the current president of South Africa. Both of them are engaged in a strange ceremony, kissing one of those humanoid called Homo naledi. And Homo naledi was a humanoid that lived in southern Africa many thousand years ago, far before Homo sapiens sapiens. The image on the right hand side shows that some of these humanoid had mixed with us, modern human, as shown by the line in blue. And we know that because in our genome today, we have up to 10% of these DNA that we call ghost DNA because we are not able to read it. As you can show in the picture, some of those admixture happen after the separation between African and non-African, suggesting that if we want to understand the extent of this introgression of these humanoid, this ghost population, we need to invest in African genome variation. Someone will say, why should we be worried about those ancient DNA? And I'll answer here using a very specific African language, which is called Sankofa. This is the image that come from the Akan language. Akan is a tribe in Ghana, and this image represents a bird picking a neck on his back, and that represents taking information in the past, and this egg will seed a new bird, and that means to feed the future. And how can we apply that to genetics? Dr. Pablo won the Nobel Prize for Physiology and Medicine a couple of years ago. And one of the reasons he won that Nobel Prize was his capacity to fully sequence the genome of the Neanderthal. But not only fully sequence it, to show the impact of those old genome on human health and disease. For example, the Manhattan plot shown on the left-hand side show on chromosome 3 an area in the genome that increases susceptibility to COVID-19 during the pandemic in people of European and Asian ancestry. Of course, the people of European and Asian ancestry, they have up to 2% Neanderthal DNA, and that happened after out of Africa. And the very same region is associated with other threat and diseases like neuropsychiatric disorder. That suggests that ancient DNA or DNA inherited for other humanity in humanoid matters, and studying African population will improve that knowledge. I always speak to the African humanity living in Africa for more than 200,000 years with wave of migration and admixture. And these also lead for some variant to be specific to African population and other variant to be much more common in African population, with implication for all of us. For example, when we published this paper during the H3 Africa Consortium project, we reported only in 500 African genome more than 3 million single nucleotide variation that was not in any reference database. And this is another example of a variant that is very common in nonsense mutation amongst African American, 200 times more common in African American than in American European ancestry, associated with 40% reduction of blood bad cholesterol for those who have the variant. And these have been used to develop specific medication. One example is shown on the bottom, and that medication to remove bad cholesterol from our blood. And this medication is actually taken globally by more non-African than African populations. The second reason, as we say, is the Africa ecology. Exposure to diverse environment have selected also very specific variants that are very common in Africa. One example is the exposure to trypanosome that is responsible to the sleeping sickness. And for that reason, that variant called G1 and G2 in a gene called APOL1 have also been responsible for increased kidney disease in population with African ancestry. And we actually, fortunately, know that the therapeutic modality targeted specifically APOL1 now in clinical trial to treat kidney diseases that will be useful not only for people with African ancestry, but also for people without African ancestry. We already know the selection of sickle cell disease because of exposure to malaria, and in this publication that we reported very recently, we map here that Many other malaria-resistant genes, also including APOL1, map the same region in Africa, suggesting that there may be some interaction between these variants that are relevant for treatment. To speak to sickle cell disease, I have argued a couple of years that we need to invest in African population to find new target for therapeutic manipulation for sickle cell disease. For example, the target that modify the level of fetal hemoglobin. The reason being that if you look at the pie chart on the upper right, this is the representation of the non-variant associated with increased level of fetal hemoglobin, including BCL11. They explain up to 50% heritability of fetal hemoglobin in people with European ancestry. The lower pie chart show that the very same three regions explain only maximum 20% heritability of fetal hemoglobin in people African ancestry, specifically with sickle cell disease, including some of the studies that we perform ourselves in Cameroon. This suggests that at least 80% missing heritability exists for fetal hemoglobin in Africa, and we can explore African genomes to reduce that level of missing heritability. We all know that the African genome representation in the global database is very low, less than 2%. And this is a problem for equity in genetic medicine. This is an example from the UK Biobank using polygenic risk score to evaluate the prediction accuracy specifically for blood threat. African populations in the UK Biobank represented here in purple perform very poorly in terms of this score of prediction. And this is because most genomes of people without African ancestry have much shorter haplotype in their genome. That means that the prediction would be inaccurate in those populations. We developed during the H3 Africa Consortium an array that we call GWISE H3 Africa GWISE array, taking an advantage 500 genomes from all across Africa to develop a GWISE array that we hope to better capture the genome of people with African ancestry. And we use that GWISE array to study fetal hemoglobin genomics in sickle cell disease from population in Africa. Our first discovery cohort was from Cameroon, and the reason for Cameroon is that, as shown on the picture on the lower right, it has a very high genetic diversity. Most people with sickle cell disease were not exposed to hydroxyurea, and the diversity also means that it can represent an optimal place to study African genome variation. What we also did, we studied sickle cell disease population from Tanzania using available genome-wide association study that was published some years ago, and also all available genome-wide association study from African American, and perform a global meta-analysis. Totally around 4,000 patients with sickle cell disease. This is the largest study of fetal hemoglobin with sickle cell disease patients in the world. What we found, we confirmed that BCL11A on chromosome 2 remained the strongest loci associated with fetal hemoglobin. We replicated also MYB loci on chromosome 6, but we discovered 14 new loci that have not been reported before to be associated with fetal hemoglobin, including FLT1 in blue, on which I'll focus a little bit for this talk. The heritability estimate before the study was around 20%, and actually 14% in amongst Cameroonian patients with sickle cell disease. By using a GWISE array that properly beta capture African genomes variation, we moved the heritability to 90% as shown here, and it remained extremely consistent despite the use of different type of principal component analysis. In this specific talk, I'll focus on a gene that we found to be associated with fetal hemoglobin called FLT1. The first reason is that it have not been identified before in a genome association study. It's novel. The second is replicate in a meta-analysis across all imputation panel that we use for this study. The third reason is that it's actually about the strongest signals after what we know, BCL11 and MYB loci. This gene is also very interesting because the minor allele frequency that we found here in homozygous state can be associated with fetal hemoglobin to nearly 25%. The current target for therapeutics is 20%, suggesting that this could be a very good target for therapeutic manipulation for fetal hemoglobin for treating sickle cell disease. Someone may ask, well, how is that we haven't found it in previous GWAS analysis? The answer is shown here on the circle in red. If you focus on the allele in blue, in red, and also in orange, these alleles are only found in population with African ancestry, and you cannot find them in Europeans, in Southeast Asian, for example. It's found a little bit amongst African American and Qatari population because of an obvious admixture of the African populations, suggesting that we would have done this genomal association on 1 million European, we would never have been able to find the association with FGL1 because the variant that we found is monomorphic in European population. What happens if we remove FLT1 in a mice model? If we remove FLT1 in a mice model, it's not compatible to the life of the mice because the mice will die very early, actually, because they don't form red blood cells. They also don't form blood vessels. We also realized that the genes belong to the complex of hypoxia. They're connected to the complex of erythropoiesis, the development of red blood cell. And it have already been shown that actually hypoxia-inducible factor 1 alpha was associated with increased level of fetal hemoglobin. So what we then do, we expose K562, which are erythroleukemia cell line, to hypoxia and look at the production of FLT1. And the picture on the right-hand side with red bar shows an increased level of production of FLT1 with increased exposure to hypoxia. And on the panel on the right-hand side in blue, we show that fetal hemoglobin start to be expressed after they fall in these cells. We also realized that the area where we found our GWAS signals correspond to an area a little bit the promoter of FLT1, suggesting that there could be a transcriptor factor target, as shown on the data from ATAC-seq data here in blue below the first panel. We took advantage of a collaboration with colleague Greg Newby to delete those areas in two ways. First, by deleting a bigger portion, and then by deleting a specific area where we know there is a target for a gene called GF1 associated fetal hemoglobin production. And this led to increased production of FLT1 by 2.7 to 4-fold, suggesting that the GWAS area that we found are probably repressing the expression of FLT1. We then inhibit FLT1 this time in CD34 erythroid cells, and the cell that actually produce fetal hemoglobin. The arrow in red shows, the first arrow shows the exposure to inhibitor FLT1 that reduces the fetal hemoglobin. The fetal hemoglobin is on the y-axis. When we expose the same cell to hypoxia, is the second arrow, the fetal hemoglobin is restored. And we found the exact same result that the second panel with F cells. So we believe that fetal hemoglobin FLT1 is associated with hypoxia-induced level of fetal hemoglobin, and we are working to understand the mechanism at this moment in our lab. So what we have shown here is that up to now, the low representativity of African genome in the global database means that we have been telling our genome or understanding our human genome with a single angle, the angle of European population genomic variation. Is just telling a single story. And like Chimamanda said, it's not that the story is not true, it's that the story is incomplete. And I have proposed that we need to sequence at least a 3 million genome across Africa if we want to meet to reach the missing gap or to understand the human genome to fully to improve not only our scientific knowledge, for example, for cure sickle cell disease, but also for the imperative of equity. And I would like to thank you for your kind attention and thanks my founder, in particular, the NIH that have supported my work for a very long time. Thank you very much.
Our second speaker, Dr. Boro Dropulic, received his PhD from the University of Western Australia and his MBA from Johns Hopkins University. He has been in the gene therapy field since the late 1980s. After a Fogarty Fellowship at the National Institutes of Health, he joined the faculty at Johns Hopkins, where he worked on developing lentiviral vectors as delivery systems for gene therapy. After 4 years in academia, he founded his first company, Virexis, and led the team that demonstrated the first safety of lentiviral vectors in humans with his University of Pennsylvania colleagues. Later, he founded Lentigen, which first developed the lentiviral vector used to produce Kymriah, the first FDA-approved gene therapy product. Later, Boro saw an opportunity to integrate lentiviral vector technology with a closed system automated cell processing device to enable distributive place of care manufacturing at hospitals, potentially improving affordability and accessibility of gene therapy products such as CAR T cells. He therefore spearheaded the acquisition of Lentigen by Miltenyi Biotech in 2014 and led the development of a global place of care network of clinical centers that were able to successfully manufacture CAR T cell products and demonstrate their therapeutic benefits in clinical trials.
Well, thanks very much for inviting us to share what we're doing at Caring Cross. Caring Cross is a non-profit organization, and our mission is really to enable affordable and accessible cellular gene therapies. This first slide is just really to orient us as to show the clinical success and regulatory approval of cell gene therapy products. And on the left there, you'll see various CAR T cell products, including Kymriah, Yescarta, for leukemia and lymphoma, and then others also for other hematologic malignancies. Then on the right there, you'll see the products for stem cell gene therapies, which an example is the Zynteglo for beta thalassemia and Lyfgenia for sickle cell disease. And there are numerous candidates in development, and I truly believe we're at the beginning of a new era of transformative therapies for patients. All these therapies have one characteristic. They're all autologous therapies. Autologous gene therapies work very, very well. And the cells are derived from the patient. So whether they're T cells or stem cells, they're taken from the patient. The cells then are transduced with a vector. We exclusively use lentiviral vectors, and many of those products also use lentiviral vectors. And then those cells, after their genetic modification, are then infused back into the patient. And on the right there, you'll see an example of a CAR T cell. The cell there is the T cell with the CAR molecules on the surface. They engage specifically the cancer cell subtype and then kill the cancer cell. But their price is very high and not sustainable and significantly restricts access to these life-saving products. And you'll see on the left there, Kymriah is $475,000 a dose. That's without any clinical cost included, just the product itself. And you can see all the way to the right, Lyfgenia, the sickle cell therapy, $3.1 million. And at such pricing, it's not sustainable anywhere for insurance companies or health systems, and certainly not feasible in low and middle income countries. And such pricing really restricts access, particularly for marginalized and underserved populations, no matter where you are in the world. And this next slide really gives you some of the data behind that. In the United States, only 25% of eligible patients receive CAR T cell therapy. And in low and middle income countries, it's less than 1%. So we're really seeing a lack of access to the patients who need it. And in many patients with some of these, the largest populations for these diseases, like the sickle cell disease, are located in low and middle income countries. So really we have a major crisis here in terms of access and affordability. So how do we, as a non-profit organization, try to tackle this problem? Well, we tackle them in 4 different major categories, if you like. One is technology innovation, where we improve the materials and the processes, resulting in products with significantly lower cost. For example, the vector manufacturing. The vector is the single largest cost of producing these products, and we've developed a new and efficient process that really significantly reduces the cost of the vector, and we pass that on to our partners. In the second column there, we not only develop the processes, but the therapeutic medicines themselves. So we've developed freedom to operate cellular gene therapies, developed with our decades of experience of the teams that we have here at both Caring Cross and our public benefit corporation, Vector Biomed, which actually makes the vectors. And I'll talk a little bit about that in just a moment. Then in the third column there, we're very much proponents of point of care manufacturing locally, because we believe that decentralized manufacturing for autologous products is a lot more efficient than centralized manufacturing. The traditional pharmaceutical paradigm is to make products and ship them out, and you get scalability and a reasonable cost when you make a product in a facility and just ship it out like a monoclonal antibody. But when you're taking cells, right, and then shipping it to facility, manufacturing, and then shipping it back to that hospital, that's very, very costly. Not just the actual transportation cost, but the actual infrastructure to maintain that logistical gymnastics. So we believe and we know that producing the product locally really significantly decreases those costs. And then finally, on the right, we're all about democratizing this technology. And so we technology transfer to certain organizations in low and middle income countries, so they can use their local labor to produce these products locally and much more affordably. This goes beyond simply the final product, but also goes into vector manufacturing and the other products that go into vector manufacturing, so they can incrementally use their skilled labor that is much less costly to produce these products affordably in their own regions. So the one important thing about point-of-care manufacturing is that you need to show reproducibility to produce these products locally. And in our history, our team's history, we've been able to be involved in a number of clinical trials. And one of the clinical trials is shown here, where we had 2 sites, one in Cleveland, Ohio, and the other one in Moscow, Russia. And we asked the question, if we would use the same device, the same materials, the same release criteria, can we produce a highly similar product? And the answer is yes. We're able to produce a highly similar product, be able to produce the product within 8 days. That means that every single patient that was enrolled was treated and had great outcomes. And there you can see in the middle there, a particular patient, a 69-year-old with follicular lymphoma, both before and after therapy. On the left there, you can see the lightly colored tumors, and then the tumors in remission here after CAR T-cell therapy. So in many cases, these products are life-saving for patients, and it's important to have a short vein-to-vein time, particularly for patients with advanced disease. If you're a patient with advanced disease and you have to wait, several weeks to receive the product, well, that's very detrimental position for the patient. And on the right there, our colleagues in Spain have already taken it to the next level. Not only do they have multiple site manufacturing in Spain, but they're also able to receive regulatory approval and product reimbursement in Spain. So very happy to see that. So this slide shows a little bit more color regarding point-of-care manufacturing. Up on the top left and right here, these are the 2 clinical centers. The one was at Case Western Reserve University, the team there that produced the product, and on the right there was the team that produced the product at the Pediatric Research Center in Moscow in Russia. And you can see there that the overall response rates were very comparable to the approved products, 82% overall response rate at Case Western, and over 90% overall response rate at the Pediatric Hospital in Moscow in Russia. And on the bottom there, you'll see a clinical trial that was performed at the Medical College of Wisconsin. And in this case, it was a bispecific CAR targeting both 19 and 20, because we know that about 50% of patients that go on single CAR COVID-19 therapy fail that therapy because of antigen loss. And in this particular case, we had a 92% complete remission rate, and after 2 years, only one relapse to date, showing the power of multiple targeting tumors with multiple CARs. And one of the products that Karen Cross is taking forward is a TriCAR that targets 19, 20, and 22 to address the issue of relapse due to antigen loss for lymphomas and leukemias. And so in terms of point-of-care manufacturing, you have to think about how to set up a point-of-care manufacturing network. And on the top left there, you'll see a typical hospital facility with a clean room and the various hoods and incubators. But if a hospital doesn't have its own facility, there are other solutions. And our biomodule is a clean room that can actually be delivered and deployed in the parking lot of a hospital. All that you need is electricity and internet. And on the bottom there, you can see the insides of that module. And so the way that we think about creating a network is you're having a reference facility where either it's a clean room at a hospital or a biomodule where you're remotely monitoring the point of care facilities, but also manufacturing product. And then on the bottom there, you have the point of care facilities having either a module or their own facility. So you have identical equipment, materials and workflow, a hierarchical system, a reference facility monitoring, a local facility reporting, and real-time data transparency and auditing. And this is what one of these facilities look like. I'm not going to go into great details, but it's a modest 700 square foot facility, which you have both the clean room to manufacture and the testing room right on the same facility. And each one of these facilities can produce 250 products a year on a single shift. But with multiple shifts, actually, because you're not using a single device, because you're using a bioreactor to put it into an incubator, you can actually scale up and even produce 1,000 doses a year from such a modest facility when you do multiple shifts. So tremendous capacity. And what that does is it lowers your fixed costs of the facility and decrease your overall unit costs for the production of these CAR T-cell products. And then a little bit about our capabilities, what we do as an organization. We're involved in manufacturing innovation on the top left there, whether it's lentiviral vector production or cell manufacturing or the materials, to developing clinical candidates on the right there. An example is leukemia, lymphoma, and multiple myeloma. And then also on the bottom, the supporting systems like biomodular implementation and then point of care logistical systems. And some of the logos there are some of our collaborators. We've developed a very economical CAR T-cell manufacturing platform where we can use either apheresis or blood. It uses the CEPAC-C Pro from Cytiva. It's a very modular process of manufacturing. We manufacture vector through Vectra Biomed, expand the cells in a G-Rex that's produced by Scale Ready, and then have freedom to operate. So we're focused on low-cost materials, vector and selection reagent, a short and simple process. Short improves the cost of goods, simple improves the robustness, decreased vein-to-vein times. It's better for patients, especially with advanced disease. The option to use blood from patients, resulting in improved access in low- and middle-income countries, and freedom to operate products, so there's no third-party IP that would affect cost. I'm not going to go into great details here, but we can use either in our process either whole blood or a leukopak. We've developed a negative selection reagent that binds the cells that you don't want, like the monocytes and the B cells, to the red cells, so that in the CEPAC-C Pro, instead of getting PBMCs, you get purified T cells all in one step. You add vector, activation reagent into the G-Rex. 1 week later, you're ready to formulate and then infuse into the patient after testing. And the total cost of this process, including the vector, is $20,000 a dose. There's a little bit here about the data showing the purity of our product when we compare our negative selection to positive selection, very, very comparable. And then we get enrichment of gamma delta T cells using our negative selection method over positive selection methods. And we get a very high proportion of the T stem cell memory cell component. You can see in yellow here, it's the bulk of the population of our final product on the bottom right there in yellow. And we also have, we couldn't rely on third parties to make vector for us at an affordable cost. So we said, well, we know how to make vectors. So we spun out our company, Vector Biomed, and it is located in Gaithersburg, Maryland. It's a public benefit corporation and has tremendous experience, and it makes low-cost vector for third parties, for anybody, but also special pricing for Caring Cross in order to support its model. And very briefly here, there's an issue with the current state of cell and gene therapy. Is despite groundbreaking therapies, it's stalled by multiple factors, including high cost of goods, limited access. And we believe the future of gene therapy is to dramatically reduce the cost of goods and provide customers and clients with end-to-end solutions. Relatively new company, opened the doors in 2023. We have a fantastic team, already 40 clients worldwide, and we believe that we'll be in a position to be a market leader in 2030. We produce both custom and off-the-shelf CAR T-cell products, what we're calling the Lentiverse, and our true platforms that scale with our partners, either custom vectors, off-the-shelf, targeted vectors, cell manufacturing solutions, and the supporting reagents. And accelerating development from concept to design to testing and identifying the most optimal constructs to manufacturing. We support our partners through the entire process, and even later, assay and regulatory support, pre-commercial support, and then commercial production of the vector. We have developed a high titer process in which we get multiple 10 to the 8 transducing units in the harvest that results in multiple 10 to the 9s in the final product for a model vector. And so I think we produce something that's very unique compared to our other colleagues in the space. Very fast production of vector, high quality and value. We're about half the price of our competitors to produce GMP vector. I want to briefly talk about our strategic collaborations. One we have in Brazil is that we are in the process of technology transferring, not just the CAR T manufacturing, but all the vector manufacturing and the materials and reagents, so they can produce it locally at about $35,000 a dose. So this is a partnership now that's been ongoing, where the Minister of Health had some very nice things to say. We are on timelines at the moment with our products, our delivery. And we've also developed a strategic collaboration in Turkey with the Koç Healthcare Group. So very excited about that collaboration. We have multiple partnerships in India with several organizations, and also in the United Arab Emirates with the Burjeel Hospital Network there as well. We're initiating a collaboration also in Nigeria, similarly with the Wellbeing Foundation, in order to bring equity and to bring this kind of manufacturing capability into Africa. We also have other initiatives, particularly with the Health Innovation Exchange, a Geneva-based group, in order to raise a fund to further improve the affordability of these products globally, particularly in lower middle income countries. And so health equity is a number of things. It's the clinical infrastructure, it's the facilities, it's the training of personnel, it's the GMP manufacturing of the ATMPs, it's the GMP manufacturing of the materials that are required for the ATMPs, it's a pipeline of ATMPs, it's preclinical development and R&D. When you create this kind of an ecosystem, that's how you ultimately get to health equity. We're also developing a network in the US, partnering with several hospitals in the US in order to make this affordable in the US. Currently, Medicare and Medicaid pays about $257,000 for the product plus the patient care. Our material cost, as you've seen, is $20,000. Laboratory and labor and facility costs another $20 to $30K. If we're making this product for a total cost of about $40 to $50K, we can make it really affordable and make it positive for many hospitals in the US. So we believe that hospitals should be able to make these manufactured products, even commercial products, when these commercial products are not available for patients for any reason that will negatively affect patient health outcomes. For example, if there's no product available, certain CARs for pediatric cancers are not available at all. So hospitals should be able to make them. Or if the delivery time is too long, or if there's cost or insurance delays, or if there's geographic access issues, in each one of these circumstances, hospitals should be able to make the product if products are not available for patients. And we have a pipeline of therapeutic products. I'm not going to go into them, but everything from a tricar for leukemia and lymphoma, for multiple myeloma, for sickle cell and beta thalassemia. And this is only possible when we're collaborating with a large number of partners. So I'm not going to go into summary here in the interest of time. And I'm so excited about this field just because it's engineering, and I think we're at the very beginning here of a future that will create these life-saving therapies that's going to get incrementally better. And finally, just to thank our partners, which is very essential to our model. Thanks very much for your time.
Our third speaker, Dr. Jennifer Adair, is a professor and vice chair in the Department of Genetic and Cellular Medicine at UMass Chan, and associate director of the Horae Gene Therapy Center, also here at UMass Chan in the United States. Jen completed her BS degree in chemistry at Youngstown State University and her PhD degree in genetics and cell biology at Washington State University. After a postdoctoral fellowship at the National Institute for Environmental Health Sciences, Jen began her work in gene therapy at the Fred Hutch, becoming faculty in 2014. In 2020, Jen was named the Fleischhauser Family Endowed Chair in Gene Therapy Translation. In 2025, she joined the faculty at UMass Chan to co-lead the first academic department focused on gene therapy translation. Her laboratory develops tools and methods for safe and effective delivery of gene therapy. Jen also co-founded the Global Gene Therapy Initiative with Dr. Sissy Kityo in 2020.
Thank you. It's my great honor and privilege to share with you the work of the Global Gene Therapy Initiative, or GGTI, today. I want to start by saying that I'm honored to represent hundreds of members from around the world who have contributed to the work, and I'll highlight some of that for you today. I think it's no mystery to this crowd that we're seeing gene therapy really come into its own. These products, also called ATMPs, or Advanced Therapeutic Medicinal Products, in particular in Europe and the US, are starting to receive tens of approvals globally. And the United States is leading in these approvals, but the majority of these products are being developed in high-income countries, as defined by the World Bank. Yet, the majority of global disease burden is borne by low and middle-income countries, or LMICs. And these are where ATMPs could have the most transformative impacts. But the standards that we have set in high-income countries make it very difficult for low and middle-income countries to participate in gene therapy development. These are complex complications involving lots of different stakeholder groups across many different sectors. And this gap is really what drove the formation of the Global Gene Therapy Initiative, or GGTI, in 2020. We are a grassroots network of members representing every sector in gene therapy development, from patients and advocates, researchers and clinicians, regulators and policymakers, to commercial investors. We meet weekly on Zoom for the last 5 years and continuing moving forward, simply to talk about what is required, what is happening in the gene therapy space, and to allow individual geographic regions to self-nominate to participate in gene therapy development. We don't presume that one answer will solve all problems, but rather engage in conversations that allow different problems to come to light and collective solutions to be proposed. We are guided by an incredible group of advocates known as our International Community Advisory Board. These are patients and caregivers from around the world. And the first piece of work that I want to highlight was our first working group report, which was published in 2021. Again, reminding folks that there isn't one answer. We did say that there are 4 key areas of parallel effort that need to happen in any geographic region that wants to try to advance transformative medicines. The first is community outreach and education to ensure that you're developing gene therapy products that patients want and therefore ensuring adoption later. Clinical readiness and implementation. Clinicians are often the first people to introduce gene therapy to patients. And we need to make sure that they feel enabled, empowered, and capable of administering and monitoring these ATMP drug products. We also know that sustainability in the gene therapy market can be achieved many different ways, and we need to invest in research and have new technology development to ensure that these therapies are developed with a local context in mind towards sustainability. And we need regulation and policy infrastructure in place, not only to ensure public safety, but also to provide a pathway for commercialization and local economic gains. And as I'll remind you, our stakeholder groups are shown in the bottom right, and we really center patients in all of this process. Last year, we published 4 years in 6 case study countries and their progress on the path to implementing cell and gene therapy. We do not go into geographic regions and ask them to take up gene therapy. Rather, we allow countries to self-nominate. These 6 countries are in various stages of implementing cell and gene therapies for which they bear very high patient burdens, but those drug products are either already authorized in the US and Europe or in late-stage clinical trials and approaching authorization. This was work that was published by a fellow from the World Economic Forum, Kevin Doxson, along with GGTI members. And I'm going to spend the majority of my time today highlighting our work in one of these 6 countries, Uganda. This is a small country in East Africa with a high prevalence of HIV infection and sickle cell disease. My co-founder of GGTI, Dr. Sisikitio Mutuluza, is a world-renowned researcher in HIV clinical trials, received one of the first PEPFAR awards in sub-Saharan Africa for HIV antiretroviral therapy implementation, and she is the executive director of the Joint Clinical Research Center, or JCRC, which self-nominated early on in GGTI's progress. As Sissy knows from her experience in the HIV field, advocacy is imperative to ensuring true progress towards accessibility and affordability. And so she was a huge supporter of bringing patients together. In this case, we bring patients who are examples of the transformative power of cell and gene therapies. Adam Castillo, aka the London patient or second patient cured of HIV infection through bone marrow transplant, and Jimmy Olegair, the 4th patient treated with CRISPR gene therapy to treat sickle cell disease, along with local advocates in Uganda, specifically Evelyn Mwesigwa Harlow, who is a sickle cell disease advocate, and Moses Supercharger, who is an HIV advocate. We also bring science to these communities. We've developed literature around gene therapy for public and media consumption, which can easily be translated into local languages and cultural contexts. And we host events with local community members to engage with experts from all over the world in gene therapy development. And probably most importantly, we elevate these patient voices in our scientific circles. This is a community piece written by our advocates, which published alongside our case study of those 6 countries in Science Translational Medicine in 2024. And our patients really advocated strongly from the beginning that unless we can screen and diagnose and easily monitor for the improvement after gene therapies won't have a place in these geographic settings. And so a lot of our work also involves technology development around affordable, accessible screening and diagnostic tools, as well as monitoring post-gene therapy. And I'm showing here just an example in the sickle cell disease space. Dr. Umit Gurkin from Case Western Reserve University and development of the Gazelle for measuring hemoglobin in the field at low cost per test. This is a device that's now marketed by Hemex Health and widely distributed across sub-Saharan Africa. And a graduate student from Uganda who is in Umit's lab, Solomon Oshabahibwa, who's developed a miniaturized microfluidic test to test for red blood cell occlusion, which is a functional output in sickle cell disease. We're also training the first generation of Ugandan gene therapy researchers, Dr. Lois Baiga on the right and Dr. Rachel Bachaita Cheune on the left. These 2 women have papers in progress right now. They've presented at the American Society for Gene and Cell Therapy. They've trained in labs both in the US and in foreign countries. Lois is now back in the JCRC in Uganda implementing stem cell gene therapy there. We've also provided training for the first set of Ugandan clinicians. Here I'm showing a JCRC physician and nurse who attended the infusion of the first patient on an anti-HIV CAR T-cell trial being hosted at the University of California at Davis. And the opportunity for these clinicians to see that very first patient being infused gave them a lot of understanding of what these clinical trial processes look like. And following this training in a high-income country, they went to India, where they saw the administration of CD19 CAR T cells to patients locally there, giving them both a high-income and a low-income country context for administration of cell and gene therapy. In terms of clinical readiness, we've supported leukapheresis, bone marrow transplantation, and laboratory standards improvement. I say we there, this was really driven largely by Dr. Kitio Mutuluza and her teams at the JCRC. Our network at GGTI simply provides connections and resources and essentially consultation around the implementation of this capacity. And in terms of regulatory process, we've collaborated across international bodies. I'll highlight just a couple of the wins in the last 5 years, including the first organ transplant bill passing Parliament in 2022, enabling bone marrow transplant to happen in Uganda. Having the president direct the Parliament to review a biotechnology policy which will govern genetic editing for both human health and agriculture in the country, which we expect to get signed later this year. And the International World Health Organization naming Uganda a test case country for the implementation of governance infrastructure for gene and cell therapy upon request. And while all of this was happening, the US was proceeding towards the approval of the first 2 gene therapies to treat sickle cell disease here in America, Casgevy and Lyfgenia. And this was a phenomenal achievement for the developers of both of these drug products, and really something that we should be proud of. But the price tags associated with these drugs are limiting, not just for patients in low resource settings who will have to self-pay to access these drug products in foreign countries, but also for the healthcare systems in the United States, has been discussed in many forums sponsored by the ASGCT. And this actually creates an interesting opportunity. As these therapies were being approved, we had multiple therapies that were not failing safety and efficacy, but were being withdrawn from the market because competition was just too high with 2 authorizations at the same time. And this means that there's an opportunity to transfer technologies that are sitting on a shelf right now that have shown clinical safety and efficacy to places where higher patient burdens exist to jumpstart gene therapy development in those locations. We started advocating for this in early 2024 as part of GGTI, and we were quickly met with the following question. While that is a great moral argument, what would be the business case for gene therapy in places like Uganda? And so in the last few minutes, I'm going to highlight some new unpublished data that's currently under review, where we've tried to establish that a business case does in fact exist. This work was led by an incredibly talented graduate student, Felipe Montano Campos at the University of Washington and the Choice Institute. Felipe is now a postdoctoral fellow at the University of Southern California. And he developed this way to establish cost effectiveness of gene therapy for sickle cell disease in Uganda by tailoring our high-income country evidence to Uganda's economic context. Cost effectiveness models are used by governments and policymakers, as well as commercial developers, to understand at what price a certain therapy would be considered cost effective over standard of care in a given space. Now, the challenge is we have lots of data around this in the US, but we don't really have any data on the implementation of gene therapy and its associated costs in Uganda. So Felipe's solution was to take the standard of care costs for sickle cell disease in the US and compare that to what we know about the standard of care costs for sickle cell disease in Uganda, and take the difference between those 2 calculations and call it a scaling factor. He then applied those scaling factors across multiple cost effectiveness models to 2 different gene therapies, Casgevy and Lyfgenia, for sickle cell disease, and established what would be associated pricing and what would be essentially the incremental cost effectiveness ratio. Now, this was possible because the underlying biology of gene therapy means that the outcomes should be biologically consistent across populations. And this is not true for all drugs, but we believe it to be true for gene therapies. And what Felipe found and presented as part of the ASGCT's annual meeting this past May was that one of these two gene therapies, Casgevy, which has a list price of 2.2 million in the US, would be cost effective in Uganda at a list price of $40,205 per drug product, as shown in the yellow circle in this table. Now, only one of the two cost-effective models met this threshold, but it's an important number to reach because it allows us to ask the next questions. Is it possible to make an ATMP for less than $40,205? Is there willingness in Uganda to pay that price? And is that price insensitive to the Ugandan economy, or would it be considered reasonable? Well, GGTI partner Vikram Matthews in Vellore, India, has published that he can manufacture CD19 CAR T cells for around 35,107 USD per product. And as shown in the square on the right, in that pie chart, the largest proportion of costs come from things that have to be imported from the US and Europe. And so local manufacturing of those components could potentially reduce costs further. We also know that Ugandan patients with sickle cell disease, who we call sickle cell warriors, and their families have paid upwards of $50,000 to $70,000 USD for haploidentical or unrelated bone marrow transplants in India out of pocket, suggesting that there is in fact a willingness to pay threshold. And finally, the World Health Organization in 2020 issued guidelines on country pharmaceutical pricing policies, where they suggested that to be sensitive, you should not exceed a drug product cost of more than 3 times the GDP per capita, and $40,205 is less than 3 times the GDP per capita in Uganda. Altogether, this suggests that there is in fact a business case for gene therapy implementation in Uganda, but it's unlikely that governments would want to send or pay money to high-income countries to do this. And so last year in 2024, advocate Evelyn Harlow and myself published this comment in Nature, where we advocated that incentivizing local manufacturing would actually allow governments to not only invest in the local health of their populations, but also reap the economic benefits of joining the gene therapy development pipeline. I'll finish by saying that in GGTI, we believe that the problems we solve to enable access to gene therapy in low and middle income countries will actually present solutions that enable access to gene therapy for patients everywhere. I'll thank you for your attention. Thank you so much for listening to our 3 presentations. Just a couple of quick announcements before we jump into our live Q&A. First is for those who are interested, I know we had a join ASGCT slide at the beginning. Importantly, ASGCT is redefining its membership levels, and effective October 1st of 2025, those of you in low resource settings, as well as students and trainees, can have access to free membership to the ASGCT, and I encourage you to join as members under that new membership guideline. I also need to say that, as you can see based on your screen, unfortunately, Dr. Boro-Dropulic was not able to join us for the live Q&A today, but we will take any focused questions for him, send them in an email, and see if we can get answers posted in the chat for you at the end. And with that, I'd like to open it up for Q&A. I see that we have a number of questions in the chat that came through during presentations, and so we'll begin with some of those questions. I'm seeing at the top of my list, is there a regulatory procedure for cell and gene therapies? Is the regulatory procedure for cell and gene therapies a regular biologics licensing authorization, or which are the fast track pathways that we can take? And Terry, I think you might be the best person to actually approach this question.
Yes. So to answer the first question, in the United States and in Europe, that I'm familiar with, and in Japan, the application is standard BLA. The availability of fast tracking depends on the nation, and there are several ways in which fast tracking may occur, particularly for those for rare diseases and through those that have what's called breakthrough designation. Again, I think that is likely to vary by nation, but one should consult with their local regulatory agency to determine whether those mechanisms are available.
Thanks so much, Terry. And from the same individual who asked the question, who are the colleagues who achieved approval in Spain? The hospital exemption approval was received by the Hospital Clinic de Barcelona, to answer that question. Ambroise, I think for you, we have a question about how can we get the scientists involved in cell and gene therapy to focus more of their efforts in African people? What is currently lacking that might help this pivot in the cell and gene therapy field?
Yeah, that's a great question. Can you hear me?
Yes, we can.
Because I cannot see the image there. I think some of the answers will come from your presentation, Jenny. I think the first thing is the knowledge. I still really think that we do not yet know our genome the way we should know it. There are still areas that are obscure to us. There are still areas that are a regulatory area, the variation, the extent of genes that can be targeted for therapeutics. And again, I'll argue that African population genomic variation is the best opportunity. But how do we get people to get involved into that? I think the whole ecosystem of research needs to be aware of it. The first level is the advocacy to policy makers. We've seen a lot of transition in terms of funding policy these days. So that type of message needs to reach those tables so that it become a priority for funding. The second layer is the scientists themselves. We need to get people who are involved in African genomic variation be trained that will move from all layers of the science education, PhD student, postdoc student, faculty who focus on African genomic variation. Another layer is communication, and that communication is formal communication, like in journals, like in Nature and so on, and get those being published in very highly visible journal. We've been lucky of doing that, but that luck needs to be distributed. Informal communication to general public, and of course advocacy is key. We need to get Minister of Health across African country, higher education, put in genomics as the center of their agenda.
Thank you so much, Ambroise. Carolyn asked, what are the benefits of focusing on lentiviral vectors, and have you considered expanding, diversifying to other vectors or delivery mechanisms? So I think that this question was focused towards Dr. Dropulich. What I can say from my experience working with lentiviral vectors is that first and foremost, they are, in terms of hematopoietic cell-based gene therapies or blood cell-based gene therapies, probably the most widely used tool to administer gene therapy to date, which means there's a lot of experience in patients with these. From a biological perspective, the beauty of viruses is that they evolved to deliver cargo into cells. And so with synthetic particles like lipid nanoparticles and other particles, those the ability to get into cells has to be engineered. And therefore, it takes quite a bit more effort to understand how they engage with cells and how they could get through. And so there are some both evolutionary and clinical experience benefits of lentiviral vectors. We will put forward the question about whether or not Caring Cross is considering expanding or diversifying to other vectors and delivery mechanisms to Dr. Dropulich, and hopefully get an answer for you. Can you speak, next question, to the strategy to demonstrate site-to-site comparability in the decentralized manufacturing model? This is again a question for Dr. Dropulich. I'll point you to a publication that they put forward where they compared manufacturing of a CD19 CAR T-cell product in Cleveland, Ohio, as well as in Moscow, Russia. And they showed there that if you use the same supply chain, the same batch record or manufacturing process, and the same equipment, that you not only get products that are comparable, but patient outcomes that are comparable as well. And so I think Dr. Dropulich is certainly one of the authors on that paper, and you can look that up. What are the key drivers for the high prices of products like Casgevy and Lyfgenia in the US? This is a tricky question. There are some high-level themes, and Terry, I'll ask you to feel free to weigh in here if you like. But generally speaking, in the US, I showed that pie chart in India where I said costs of goods that had to be imported from the US and Europe were the highest contributors to that price tag for CD19 CAR T cells in India, which means that US and European scientists are also paying those prices. So the cost of development is higher, and the cost of research and development has to be recuperated in the price. But generally, the price of drugs in the US is based on a value-based pricing model. So it is around how much value the treatment provides to the patient and to society, as opposed to what would be like a cost plus model where you take the manufacturing cost plus some profit margin. Terry, would you like to add anything else?
No, I think you've really identified that the research and development costs and the risk associated with research and development, since there are often research and development efforts that go on in many parallel programs to narrow down to one therapeutic product, that commercial manufacturers don't have another way to recoup that. However, once the technology is stabilized, you're back to the cost of goods, as you mentioned, and those certainly do vary themselves by country, related to things like labor, shipping costs, etc. Also, the facilities costs are substantially higher than the facilities costs that you showed, or that he showed, having a clean room, a 700 square foot clean room in a hospital or a biomodule. That is a very substantial change, and or difference, and the staffing of U.S. facilities, European facilities, has to be maintained all year round, regardless of what the actual need is. So there's a tendency to only develop that when in an individual company when there's enough funds to support it.
Thank you, Terry. Ambroise, you mentioned that we need to sequence 3 million African genomes. Is there an estimated cost attached to that number of sequences being targeted?
Yes, I think the first is that why 3 million? What we know from the Human Genome Project is that we have 3 billion nucleotides. And from one person, from me to you, Jennifer, we have 1.3, 1,300 nucleotide difference. Okay, if we estimate that each African have at least one nucleotide that is different because of the high genomic variation, we need 3 million Africans to be sequenced. And that 3 million cannot be in a single country. It has to be diverse according to geography. It has to be diverse according to ethno-linguistic origin. At the time we published that estimate in Nature about 2 years ago, the cost was $1,500 per individual. That include the cost of sequencing and minimum phenotyping, and that cost was estimated at 4.5 billion. I believe with the cost of sequencing today, that cost can be dropped by one third. That would be 1.5 billion. And actually, we currently, and a few others, organizing a project called the Center of Excellence for Genomics in Africa, where we intend to establish 10 centers that will be costed at 10 million per year to achieve that goal. And we are targeting around 300,000 to start for the sequence of 3 million. And then we are having already some traction to gather some resources. It's highly likely that in the next couple of years, we will see the establishment of GenCo to achieve this objective.
Thank you so much, Ambroise. I am seeing that we don't have any other new questions in the chat, unless I'm missing one or two. I would like to say I have one more question for Dr. Wonkum. Ambroise, with the FLT1 target that you identified, what do you see as the path forward to clinical translation of that as a gene editing target for the treatment of hemoglobinopathies, including sickle cell?
Yeah, that's as you know, Jenny, this is a long way forward. I had a great talk with Dr. Stuart Hawkins, who described DC11A. And he told me in the course of publishing this paper that he took him from the time of gene discovery 12 years to understand the mechanism.
Mm.
So yes, working very hard to understand how is it regulated fetal hemoglobin. So that's probably the first things that we're doing. It's happened that the expression or the production of FLT1 is very transient in CD34, so it will not be an easy task. So we are using K562 as the model, but we know it's not the right model. We try to use UDEP2, but UDEP2 does not produce endogenously FLT1. That means that we need to go back to CD34 and find the right way to understand that mechanism. What will be the time to translation? I don't just know. It will depend on the speed of our research to understanding the mechanism, but it also depends on our luck to find the mechanism. There is a combination of two.
Thank you so much. I think I just want to check in that we do have one question left in the chat. You mentioned that the FLT1 knockout is incompatible with life. We know mouse development is not the same as human embryonic development, especially in terms of erythropoiesis. Has there been any studies to try and rescue these mice with human FLT1 or any early erythroid transcription factors to better characterize the mechanism?
That's a great question. And no, that study have not yet been done. Of course, FLT1, we published it one early this year, so we haven't yet looked in the way we could at least study mice that are heterozygous or hemizygous of FLT1. That's the first thing to do. The second is that embryonic development in mice, specifically fetal hemoglobin, does not work like a human. Mice doesn't do fetal hemoglobin, so we will need to have humanized mice to start to explore such challenges. Yes.
Thank you so much for that. And thank you for attending today's Around the World webinar series. Again, we look forward to inviting many more members and continuing this series moving forward. Thank you so much. Dr. Flott, I'll hand it off to you.
Well, thank you, Dr. Adair, and my thanks to all of you for attending today's webinar. I want to also give a special thank you to our speakers for sharing your expertise and for a great discussion. This event was recorded, and all attendees will receive an email with a link to watch the recording when it's available. I'd like to also spotlight two important upcoming events. First, the Advancing Cell and Gene Therapies for Cancer Conference in Philadelphia. We've extended our abstract deadline to Friday, August 22nd for this event. This is available both in person and on demand. If you're working in cancer research, we encourage you to submit an abstract on your work before the 22nd, which is just in a few days. Second, don't miss ASGCT's unique policy summit on September 25th and 26th in Washington, D.C. This exclusive annual event brings together policymakers, federal regulators, cell and gene therapy professionals, scientists, and patient advocates for critical discussions shaping our field. For more information and registration, please use the links provided. Finally, ASGCT values your feedback and welcomes suggestions for our future sessions. Please take a moment to complete the survey linked on the right side of the screen under evaluation. And once again, thank you.